But yeah. I just want to get rid of it. You guys see my cursor? I want to click on the OK. There we go. I've never been so lost. Sorry. Oh, it's my video. Okay. Cool. Very good. Sorry about that. Yeah, hi everybody. Thank you for coming. Hector, thank you for a great talk. So my name is George Kanaan. I'm in Jody Deming's lab. I'm a PhD student in oceanography and astrobiology, obviously. So today I'm going to talk to you about the detectability of sea ice bacterial polymers in simulated ocean world plumes, and we're going to break that title down and step into each one of those terms and see what I actually did. This was an astrobiology rotation done with Dr. Marc Neveu at the NASA Goddard Space Flight Center, and it was a lot of fun to be in DC for a couple months. So just to give you a broad outline of what I'm going to talk to you today about, we're going to start with motivation. Why did I do this work? Essentially, why pick sea ice bacteria as an analog in this work, and specifically why they're polymers. The hypothesis I tested, how I tested it, and then we're going to go over some preliminary results. So let's just get started. So what motivated this work? Basically, ocean worlds have become more and more interesting. The last decadal survey highlighted an Enceladus orbital lander as a not first priority, if I recall, but a second priority mission. And these icy moons are essentially kind of the next set of targets that the global astrobiology community has in mind to study and hopefully find signs of life. So as part of that global mindset and effort to find life, there's a bunch of missions ongoing right now. So, and that did already occur. So you might have been familiar with Galileo and Cassini. Cassini is a satellite that specifically generate data about the plumes on Enceladus. So for those of you who don't know, Enceladus is a moon that is essentially an ocean world that has a rocky core and a very thick icy crust on top of that ocean. And a unique feature of it, or perhaps not so unique, is that in the southern pole, there are plumes of material that are ejected from the subsurface somewhere into space. And one hypothesis is that some of that material originated in the ocean. So it's a easy way to access and understand the composition of the underlying ocean. Cassini's flavor of mass spectrometry, generated data about the composition of these plumes, and that was published by Postberg et al. In 2018. And they found a non-negligible quantity of macromolecular organics. This figure is taken from their paper. Their idea for how those organics kind of turned up in those icy crystals, how we detect them, is that you have these, basically, there's a salty ocean at the bottom, there's an icy crust. Somewhere in the icy crust, there's a fissure of some kind. And by some mechanism here, they suggest gas bubbles lined with organics, particular organics. You might get a buildup organic solid at the surface. I kind of think, as a microbiologist, I think of this as a biofilm, but I don't think they intended to be necessarily a biofilm, just a thin layer of organics on the top of at the interface here with the vacuum, so semi-Kazan vacuum. And essentially, some of that organic matter is aerosolized along with water and then ejected into atmosphere, and that's how we would have been able to measure it. But this hypothesis and model not entirely, but relies substantially on particular organic matter. So one of the things I was interested in is figuring out if dissolved organic matter would be detectable by upcoming missions, such as the Europa Clipper, which actually has an instrument called I forget what it stands for, but the SUDA, which is going to analyze dust grains using mass spectrometry, and perhaps will also find organics. So I need to clarify here that I'm going to talk and interchange sometimes about Europa and Enceladus. So right now I was just talking about Enceladus, but Europa Clipper is obviously a mission that's going to Europa. Europa is thought to maybe have plumes, but it's not confirmed. So that's why this study is kind of applicable to it. And Marc likes to think about Europa a lot, so we're in the realm of both moons, essentially. So that's kind of the broad scale motivation of what we're studying in the solar system. The very specifically though, we'd like to kind of anticipate that data that something like Europa Clipper is going to generate. And what I mean by that is that if we have results for what we expect to see, then we can be more confident about our interpretation of what we end up actually seeing generated by Europa's mass spec or any other future mission. And then a secondary goal is to hopefully understand the physical mechanics of detectability of these dissolved compounds. That just means which compounds become detectable and which are detectable from the original blank, and which aren't, and what quantities does the composition change, does the structure change, etc. So to get at this question, I picked C. bacteria. I'm biased, obviously. I study C. bacteria, and that's one of the things that made this project possible for me. But they are a good analog. Why? Because C. bacteria are essentially adapted to live in an environment that is not. I would go as far as to say that is plausible to exist on Europa and Enceladus. That is, that while they're accustomed and they occur in a pelagic environment, in the winter when the ocean on our planet, the poles of our planet freezes, they're taken up into sea ice, and they spend sometimes many years within brine pockets, within very salty pockets of water within the ice, essentially making do. And so that's really the environment they're adapted to. That's the environment where they have a competitive edge over the remainder of the pelagic community that they were frequenting in the ocean before they were entrained into the ice. So concretely, what that means is that they are adapted to very low temperatures. Metabolic activity has been measured down to -20°C, and it's thought to be even lower than that. Growth has been measured down to -15°C. They're adapted to very high salinity. So here I quoted 24% by weight, just to give you an idea. The ocean is at 3.5%, and it's kind of saltier, large areas of the ocean, but the Arctic is actually fresher at 3.1 or 3.2. And 24 is not really even the highest number. It's kind of hard to figure out what the exact highest would be. I guess it would be the detected point, but this is a good average for what you might see in the winter these days or throughout the last decade. And so this is the salinity at -30°C. The pH values in brines is slightly higher. It's actually not that well constrained. I thought it was better constrained, but when I was reading about this, I was kind of surprised. There's not that many measurements because, as you can expect, you can't really stick a pH probe through ice into tiny brine pockets. So there are some measurements though somehow, and they basically fall to be slightly more basic than our ocean. They tend to be, the values I saw were at kind of 8.5-ish. And our ocean, I think, is at 8.1. Like, I forgot. Something like that. It's more alkaline, more acidic, sorry. And that number for the pH of brines comes from Gleitz et al. 1995. So you can see how old that measurement is. The reason I point that out is because the ocean on Enceladus is thought to be slightly more basic at a pH of somewhere between 9 and 10, if I recall correctly. Lastly, these bacteria are adapted to an environment where, while there is light, they don't necessarily rely on it. The community, in some parts of the ice at the bottom, there are algae, and so the community as a whole might rely on light. But within the ice, they are consuming organic, and so that's relevant because the ice sheet on the ice crust, rather, on Europa and Enceladus is thought to be kilometers thick, so you're probably not getting a lot of light, if any, through. And so it's just another point in common that these bacteria might have with eventual life that we might detect within those crusts. And then lastly, and very relevant to this study, is that these bacteria they've evolved to strategies that involve the production of very specialized compounds to live in the ice. And so those are the kind of class of compounds that I'm going to focus on today. I'm going to broadly refer to them as just extracellular polymers. And so that's really any polymer that the cell produces and lets releases freely into the environment. To give you an idea, that those can be we're going to talk about those, but those are only one class of specialized things that you need to live in ice. Everything in these bacteria almost is optimized for life in ice, be it the enzymes they produce, the proteins they produce that have to be more or less flexible at different temperatures. They have strategies to deal with the high salinity by either taking in salt or producing salt-like particles that kind of balance that osmotic pressure at the cell membrane, which we call compatible solutes. But as I said, I'm going to focus on those polymers. So, as I mentioned, the polymers I'm going to talk to you today, they're a broad class of polymers. They're hallmarks of sea ice bacteria and adaptation to the ice. What I mean by that is that sea ice bacteria are by no stretch of the imagination the only bacteria that produce extracellular polymers. But within the ice, you find it in very high quantities, partially because algae in the ice make a lot of them, but also because they play a very unique role in the ice. They take on these specialized functions, like the fact that they act as an antifreeze, they physically alter the environment in the ice. And so what I mean by that is illustrated very well by this picture, which is from Kremsen et al. 2011. So this is work generated by my lab, which I'm very jealous of. I wish my work was this cool. But basically, what you have on the left column here is ice, salt water that was frozen right in the lab with no EPS, and here with EPS. And what you can see very clearly in these two panels is that the shape of the brine pores, of the resulting brine pores, is drastically different, right? And with no EPS, they're very regular Euclidean shapes. And with EPS, you get this kind of more matrix-like network interconnected brine pores. And you can see why this is more advantaged environment to live in than this, because you can benefit from that interconnectedness to take up nutrients that are a different part of the ice. If you're lower in the ice, you might have influx from the ocean, and if you're in the middle or the upper part of the ice, you can form a broader community that remains interconnected and can cooperate on different things. So this is completely done by the polymers, by a subset of the polymers that are produced by sea ice bacteria, and that's just one function. Another function, as I mentioned, is the antifreeze, right? It reduces the temperature at which water freezes, and so it allows for a liquid space to exist in the ice at lower temperatures than it should. And so that's critical for bacteria, right? That you need to be, even though some might survive staying dormant in solid ice, very, very few. If you want to drastically increase your chances of survival, and if you want to have any semblance of metabolic activity, you need liquid water. So that's achieved. But it also has these more generic functions that you find across different types of bacteria, not just in sea ice, but you have an affinity for metals. So some of them preferentially bind metals, which are required for different parts of metabolism. They form a matrix or kind of a base for biofilms, which are essentially these communities bacteria that are fixed or surface or an aggregate, and that confers a lot of advantages to the community as a whole. It acts as an emulsifier. There's tons of these. Just to quote some of the citations for where this comes from, Marc et al. 2009 shows cryoprotection. Krems et al. 2011, as I mentioned, show the shape of the structure in sea ice. Ewart et al. 2011 shows selective retention of EPS in ice. So actually, when the ocean freezes, that EPS is found at a higher concentration in the ice than other molecules that were present. So it shows, I mean, it's amazing to me. Casillo et al. 2015 and 2018 show that antifreeze function, and they even mention ice recrystallization inhibition, which makes sense if you think about it. An ice crystal, if you're the size of a bacterium, is like a massive knife. You know, it's going to come here and tear your cell membrane open. So you want to avoid ice crystals forming near you or inside you. So if you produce a compound that kind of prevents that from happening, that's great. And so the connection to astrobiology is that these compounds might be maybe not in an exact structure and composition that they occur here, but you might need similar compounds to survive in a similar extraterrestrial environment. That's the logic. So we're using these as analogs for what compounds, what dissolved organic compounds might occur in the brines within the crust and in the upper layers of the ocean in Enceladus and Europa. So this is the hypothesis. Dissolved extracellular polymers from sea ice bacteria will be detectable in simulated Enceladus plumes using techniques similar to those planned for future missions. So that's just reiterating what I said. We want to test detectability of these polymers. So how do we do that? The first step is to collect EPS. So very briefly, you grow a whole bunch of bacteria, liters and liters of them, and then you do ethanol precipitation, which is basically just adding a lot of ethanol to get a 75% solution of ethanol, 25% culture. Everything that's not soluble in ethanol precipitates out. You centrifuge that, collect the precipitate, dry it, and you basically have a lot of different compounds, of which many of those are these polymers. We didn't really work too much to isolate polymers because, as I said, they're a broad class of compounds. Each one would need a very specific isolation, and frankly, we're happy to test as many different compounds as we can through these methods. But generally, most by mass, the highest amount of the largest class of compounds that you're going to precipitate are polysaccharides in these cultures. We dry those. Now we have a very fine powder, like dried clay, essentially, and we want to redissolve it into a solution that's going to mimic the environment on the ocean of Europa in this case. So we dissolve them at known concentrations within a 1% sodium chloride solution that we brought up to a pH of 9 with sodium carbonate. And we made solutions at different concentrations of EPS to match basically kind of projected differences in cell density. So we know how much EPS we got from a culture. We know how many cells were in there. So we have an EPS per cell number. And so we just wanted to make solutions that on a ladder of that gradient, essentially going from ocean-like densities lower and higher, just to get a bound of how many cells kind of we would be detectable for them. Then we basically inject this into a vacuum before doing mass. So I'm going to show you what that instrument looks like. And this is where the rotation with Marc was indispensable. He has this instrument at Goddard and at the University of Maryland to kind of switch over. So forgive me turning my back to you, but this is basically, so this is the whole instrument, this is the whole setup. And I would sit here on my computer for hours on end just monitoring it. This is the solution. And then this is a metal line that basically connects to the vacuum chamber. I should mention that there are no organics in the setup, meaning that there's no kind of elastomer, seals, or anything. Everything is metal. They're copper gaskets. So there's basically no organics contamination, or very little organics contamination. You have a low flow valve here and then a high pressure valve here. and then you've got the port, and this is the chamber. This thing's totally awesome. This is it's basically like a cold, he calls it a cold finger. I forget what the correct name is. It's a copper plate that you can control the computer, and basically gets down to like minus 260 Kelvin, perhaps. No, sorry, not minus 260 Kelvin. Rather, rather it gets down to like 50 Kelvin. So yeah. And you can see the temperature go down. And it gets really cold. It's really cold. And the reason you need, well, I'll go through why you need it, but so that's the first step. Second step is you got a vacuum pump here, and then you have you can't really see it, but there's another smaller pump somewhere in the back there that maintains a very, very low pressure, which you can read here. This is the temperature of the line, all good stuff. This is the inside of the vacuum chamber as we set it up before an experiment. So you've got your injection line here, you got collection cups here and here. So the thing to know about this is that when your material is injected, it's kind of spattering all over. So it's a bit of a challenge to, it's not a fine stream. You get a lot of deposit actually within the line at you're right at the interface along the vacuum, and then get some falling off here in your collection plates. And then we put a large vacuum grade aluminum foil here to basically try to collect most of the material. Oh, cryocool, it's cryocool. And that's the copper plate I was talking about. That's the cold finger. And then there's this little window so you can look at it when you're bored. A pressure sensor, temperature sensor. This is a diagram schematic of it. So everything I was just talking about, but here I like this because there's some pictures about what the collection looks like at different stages. So, first valve, you might have or you might not have some material, second valve, same thing. And then as you get to the port, you're guaranteed to have material here. And you can see it looks like salt crystals, because basically that's kind of what it is. It's mostly salt crystals, really. And you collect it, cleanly, I would say, with metal tools into those aluminum collection cups, and then you put them in a sterile vacuum tube, and then you send them off for analysis, essentially. So the thing to note is that the pressure gradients exist, right? It's not like it's being sucked into a vacuum, and then here the vacuum is very low, and then as you go further, the vacuum is non-existent, and obviously your atmospheric pressure. So that's why you get kind of different amounts along the line. For me, just because as a terms of practicality, I just collected things that were past the shutoff valve, which was most of the material. So this is what some of the sample looks like. Curiously, actually, this one, we had some liquid water. So it's curious because basically what happens to the sample when it's injected into the vacuum is that most of the water sublimates, it's pumped out with those vacuum pumps. And then some of it is kind of retained among the crystal matrix somehow. I'm not a physicist or a chemist, but and then some of that will evaporate because the surface of the vacuum chamber is actually at room temperature, right? It's in contact with the room. It'll evaporate and it'll condense. That vapor will condense onto the cold finger. So that's why you need that there to maintain a very, very low vacuum. And we actually thought that maybe some water here is actually being kind of retained by a matrix of polysaccharides, perhaps. This only occurred in a couple of our samples, but I thought it was cool. I'd like to show you pictures of it. But most of the sample looks like that, these crystals. And then this dry, these dry deposits. So the next step was to analyze these samples. The method I used was gas chromatography-mass spectrometry. So I wasn't familiar. I heard of it and I kind of knew the results you get from it, but I wasn't familiar with how it worked until this. I'll give you a quick understanding of it. The first step is derivatization. I had to practice that word a lot before saying it up here. Basically, you have to prep your samples and your compounds to be compatible with the gas spec. I don't understand the chemistry more than that. That gives you essentially sugar monomers in our case. And then a carrier gas, which is injected through the injector and in the column, basically carries your monomers along with it through this column. And different types of molecules interact for different amounts of time with the column. So when they come out, for a specific window of time, you basically get everything that interacts with for the same amount of time with the column, which is essentially one or two compound, one compound, one monomer. You go through an ion source, which basically ionizes the sample, and then it gets analyzed by a mass analyzer, which separates the compounds by their mass to charge ratio, and then you get a mass spectrum, which I'm going to show you right now. So this is a mass spectrum, basically. So what you have here is retention time. So remember I said that they come out at a different amount of time from the line. So this is basically a feature of what compound it is that you're measuring the intensity of. And to give you an idea, this is a blank, just to control the technical setup. So there's some butyl citrate, some tributyl citrate. And this is the original EPS, not a solution, but really that powder that was just directly derivatized in whatever solutions they require in that chemical workflow, essentially. And so this is kind of the base composition that we're working with for EPS. You've got some diachloronic acid, glucuronic acid, inositol, glucosamine, and then our two contaminants. These four compounds are, to my knowledge, not novel for EPS. They're known to be in there. Inositol was, I only saw one mention of it in the literature, and it's in a pretty high amount, so I thought that was curious. But so now we're looking at, so those were our kind of blanks. Now we're looking at the actual experiment. Here you have the solution that was, the solution that was before being injected, right? So this is the solution, and then this is the injected, those deposits. So the injected solution. And what you can see is that basically there's very good agreement with what you can detect. This is a very preliminary results. The derivatization needs to be optimized to get a wider understanding of the amount of the different monomers that could be in there. And we also need to do some work on tying kind of intensity to abundance roughly. But yeah, basically first results, literally very first results that I got a week and a half ago suggest that they would be detectable by a mass spec. And so that just, to go back to our original question, what that means is that you don't necessarily have to have particular organic matter to get a detectable quantity in those crystals. Now, obviously, there's the question of, I had quite a few milligrams of material here. How much are you going to get in the plume? There's all those types of questions, but just as a baseline, seems like it's detectable. So some of the places we'd like to take this, there's a lot more samples. As I mentioned, we have all those different concentrations of EPS that need to be analyzed. We would like to understand whether certain polymers are more or less retained or more or less detectable and retained in the crystals. And the reason we like to do that is really this point is because we'd like to figure out if basically there are some kind of chemical signatures of these polymers that allow them to be more retained, that are tied to their function, because then we could make we could pose a hypothesis, which is that you require these same kind of chemical signatures to get the similar function in an extraterrestrial environment, and thus the chance of it being detectable is higher. That's kind of tying that all together, essentially. And finally, we might try this on environmental samples directly from sea ice, but we'll see. With that, I know I've been a bit long. I want to thank you all for listening to me. I want to thank the Astrobiology program for funding this work, Goddard and the University of Maryland for hosting me, and of course, the Marc Neveu lab, Myron Nassim, was a lot of help with understanding the instrument. She's a grad student in this lab doing great work. And Zeming Yang is our collaborator with the GCMS, and Jody Deming, my advisor. Thank you. Geos, yeah, we're a little bit over time. We can take a question or two. Yeah, I mean.
Do you have an idea with the experimental chamber how fast the liquid were injected?
Yeah, that's a very good question. Yeah, so Fabian asked if I had an understanding of how fast the liquid was injected into the vacuum. I don't, Mark does. That number is somewhere in my computer, but I don't remember it. It's not that fast. And it's very variable, but most of it is deposited immediately after the line, so it can't be that fast. And I know you're thinking of that because of the speed of the plume injections. He does have another instrument, so I'll give him a good plug here. He has another instrument called the pipes, which is gonna solve that by accelerating particles through a magnetic field to high speed and then collect it on a cup. So that will really simulate what you might see in a mission, basically. Yeah. So yeah, that's a good point. Here we're really just testing vacuum and detectability. Yeah.
Do you think the CI bacteria polymer can get it into the air over the Arctic? Do you think your method would be able to detect this kind of like airflow?
That's a great question. There is, yeah, sorry. Yeah, Yuk asked if these polymers or other polymers produced by CI's bacteria in the ice might be detectable in the air or the atmosphere above the sea ice in the Arctic. I wouldn't be surprised if it was, because when the ice forms, you get kind of the formation of a brine skim on the top, and that can get you can get some water and dissolved compounds aerosolized by wind. I don't know. I mean, I don't know that somebody has done that, but it really wouldn't surprise me. The quantities wouldn't be substantial though. I just the raw quantity of water is a very, very thin film. Yeah.
All right, great. Well, why don't we wrap it up here? And say thank Gio again for your talk. Have a long weekend, everyone. See you next week.